Today to today's seminar, Dr. Dani Bassett. This seminar is actually sponsored by the Ludmer Center for Neuroinformatics and Mental Health. I'm just going to take a minute or two and do a bit of an introduction for what is the Ludmer Center. Ludmer Center for Neuroinformatics and Mental Health at McGill University has 4 strategic pillars: advancing big data approaches to brain research, ensuring global access to multimodal data, neuroinformatics tools and infrastructure, leading transdisciplinary research in normal and abnormal brain development, training neuroscientists, and includes 13 researchers from all career levels. So today, of course, we are welcoming Dr. Dani Bassett, the J. Peter Skirkanich Professor at the University of Pennsylvania. Dr. Bassett has a BS in physics from Penn State University, a PhD in physics from the University of Cambridge, and she's a UK Churchill Scholar and an NIH Health Sciences Scholar. She has won numerous awards. I have just listed a few of the more recent ones here, but they go sort of every year. There's a major award from the beginning of her career until today. She's the author of more than 350 peer-reviewed publications, thousands and thousands of citations, numerous book chapters, and teaching materials. And her work is supported by the National Science Foundation, the National Institutes of Health, the Army Research Office, the Office of Naval Research, the Department of Defense, the Alfred Sloan Foundation, the MacArthur Foundation, etc. She's received awards, as you can see on the slide. I just stopped sharing, from the Organization for Human Brain Mapping, and has been doing some extremely exciting work on brain networks and how they're regulated. And we are just so happy to have her here today, speaking in our seminar series on behalf of the Ludmer Center. So please go ahead, Dr. Bassett. We are very happy to have your presentation here today.
Wonderful. Thank you so much for the invitation to speak with you all today. Someday it will be in person, but for now, this is a really wonderful opportunity. So thank you. So the talk title that I gave for today's discussion was "The Brain's Structural Connectome is Organized to Support Efficient Control of State Transitions." And I know this is a little bit of a mouthful, so I'm going to be walking through each of the pieces of this title as we go. So don't worry if it doesn't make any sense right now. But what I wanted to do to kind of motivate why I want to think about state transitions for a complex dynamical system like the brain, I wanted to think about human cognition and some particular characteristics that human cognition has and that we care about, that help us go about our day. And in doing so, I want to underscore the fact that efficient control of state transitions in our brains is central to the complex cognition that we enjoy. So when I think about complex cognition, I often think about executive skills, and these include focus and attention, our ability to plan and organize, our working memory and metacognition, our ability to have initiative and persistence, our flexibility and problem-solving skills, and our ability to inhibit inappropriate behaviors or to control our emotions. So more generally over the next couple slides, and actually throughout the talk, I'll be using the term executive functions to refer to this set of skills. So executive functions are a set of cognitive processes that are necessary for the cognitive control of behavior, selecting and successfully monitoring behaviors that facilitate the attainment of chosen goals. Some common examples would be task switching. So let's say you're working on a little bit of homework and your grandmother calls and you have to switch from the mathematics in front of you to social interactions. That is a case of task switching. Or perhaps another example of an executive function is working memory, another is decision making, and another, as I mentioned before, is inhibiting inappropriate behaviors. So when we think about these kinds of executive functions, I want to posit that they require carefully controlled state transitions. So to give you a little bit more of a sense and intuition for why I think that is the case, I want to go through 3 examples of executive functions and show you how each of them requires a state transition. So for example, let's think first about decision making. In decision making, the first state of the mind would be perhaps weighing options, criteria, and values. And the second state would be deciding on a course of action. Or let's consider task switching. So state 1 is going to be task XYZ, maybe that's doing the mathematics homework. And then state 2 is going to be task, let's say ABC, and maybe that's talking to your grandmother on the phone. So this requires a switch between 2 states. And then finally, for behavioral inhibition, perhaps the first state is the urge to engage in a particular behavior. And then the second state is the inhibition of the behavior that you've deemed inappropriate. So throughout each of these executive functions, carefully controlled state transitions are required. So how do these state transitions happen? That is precisely the question that I want to address over the next 45 minutes. So first, I want to develop a theory for state transitions and their control. This will be built on a paper that we published in Nature Physics a couple years ago, and I will be focusing on the concepts in the paper, less so the mathematics, although I'll put a few equations in. But if you're curious about the details, I'm happy to discuss it further in the question and answer period. In the second section of the talk, I want to use that theory to probe normative development. So as kids grow from childhood through to young adulthood, I want to understand how state transitions may be eased to support executive function. And in the last section, I want to use the theory to probe executive function and dysfunction in the specific context of schizophrenia. So to get started, I wanted to begin by reading you this passage from Aristotle's Metaphysics in Book 12, where he writes, "Mind thinks itself, because it shares the nature of the object of thought, for it becomes an object of thought in coming into contact with and thinking its objects, so that mind and object of thought are the same." And I really like this passage because it makes me think a lot about how the mind represents the world around us and how those representations allow us to move between different cognitive processes. So we can ask ourselves, yes, mind becomes matter and is matter, but how does mind do this sort of becoming? Well, I think that mind becomes as the brain represents objects, events, experiences, and concepts. What are these representations? Well, typically what we do is that we think about representations as a pattern of activity across neurons in the brain, or perhaps across large-scale brain areas, so that one representation, let's say of a particular mushroom here on the left-hand side, is its representation is a pattern of activity where some units are highly active and then other units are not particularly active. And if we were to represent a different mushroom, this one that has these big white spots, then we would have a different pattern of activity across those same neural units. And here's just a third example, a third mushroom, a third pattern of activity across those same neural units. So these representations I will be calling states. This is a state of activation across neural units. Now, if we think about states in this way, then when we think about state transitions, we're thinking about the capacity to undergo state changes. And that capacity provides us as humans with very flexible, actuatable function. We can think about this same capacity in many other systems, but I'll be focusing on neural systems today. So then the key challenge in this space is to understand the magic of state change. How does one state transition into another state? What are the constraints on this process? To make that question a little bit more concrete, we can ask, what specific features of neural systems constrain which of these transitions can happen at all, and which just can't happen. But we can also ask what specific features of neural systems might constrain which of these transitions are easy for us versus more difficult. And when we think about the ease versus difficulty of a state transition, we have to think a little bit more precisely about the substrate that the brain uses for these kinds of changes. And that substrate is a networked substrate. So when we think about neural units, they are not independent from one another. They're all connected to one another. So every neuron is connected to other neurons. Every large-scale brain area is connected to other large-scale brain areas. And so when we think about state transitions and their ease or difficulty, we have to think a little bit more about what substrate allows for the states to change. So to do that, what I'm going to do over the next few slides is to show you a brain state, which is a pattern of activity across neural units. And that's this vector up here that I'm showing you in different colors. And on top, that's going to be on top of a network that connects the pieces of the brain together, whether those be neurons or large scale areas. So I want to think about state network pairs when I'm talking about ease or difficulty of state transitions, rather than focusing just on the activation alone. And the reason is the network underneath of it constrains which transitions can be easy versus difficult, as we'll see in a few slides. So for this particular pattern, I want to point out that the first 3 units are active, so that's the darker purple color. The next 3 units are not particularly active, that's the light color. And then the last 4 units are very active, again, the dark purple color. That state is sitting on top of a network where the first 3 units are connected in a triangle, the second 3 units are connected in a line, and then the 4th final units are connected in a square. So for neural systems, and actually for other systems that where these tools have been applied more recently, we know that the units can spread activity to other neurons or other units around them, and that having an active population, therefore, is easy if the neurons in the population are connected. So long as these units are connected with one another, you can pass activity from one to the other and have a full set of active units. And that fact helps us to think carefully about what an easy state is and what a difficult state is. So I have terms here, easy and difficult, and I'm going to try to explain why I'm using them and give you an intuition behind that before I show you a little bit of mathematics to quantify it. So I want you to focus on the top left of this slide first. What you can see is a new state where the first 5 units are active and the second 5 units are inactive. And that state is sitting on top of a highly modular network where the first 5 units are densely interconnected with one another. And then there's one connection over to the second 5 units, which are themselves densely interconnected with one another. So these are 2 modules connected by one line. So we call this a modular network. Now, this state network pair is easy because as soon as you activate one of the units in the first module, that activity can spread across the whole module, but doesn't necessarily have to transition to the second set, which could be fully inhibited. And that can be allowed by just ensuring that that one connection between the 2 modules is an inhibitory connection. I want to contrast that state network pair with something that's more difficult. So focus on the bottom left here. Here I'm showing you the same state as before, so activation of the first 5 units and deactivation of the second 5, but I'm showing you that state on top of a very different network. Here, those 2 sets of nodes that were modules before are now densely interdigitated with one another. There are many connections that move from or that link one module to the next. Now, what does that mean for the ease of holding this particular state? Well, it means it's actually very difficult to hold that state because as soon as you start activating the nodes in one module, that activity flows along any of these many connections over into the second module. So creating this, ensuring that this one is active and this one is not active would require many inhibitory control signals along every single one of those intermodule edges. As a final example of a state network pair that is difficult to achieve, I want you to focus on the bottom right-hand part of this slide, where you can see the same modular network that we had before, but now we're trying to attain a different state on top of it. And that is where we have one unit active, the next inactive, then active, inactive, active, inactive. So it's sort of a binary state. And this is a very difficult state to maintain on top of this modular network, because as soon as you've activated one of these neural units in the module, again, it can spread that activity to the other 4 units. And so in order to keep one on and one off, you have to have inhibitory control signals and excitatory control signals on almost every unit inside of this network. So that gives you a little bit of an indication for what I mean by easy versus difficult. What I mean is the number and extent of control signals that would need to be injected into the system to allow that state network pair to exist. And that same definition of easy versus difficult related to the number and extent of control signals helps us to think about which state transitions are easy versus which state transitions are difficult. So in other words, this doesn't just help us to understand single states, it also helps us to understand state transitions. So here in this slide, I'm showing you an easy state transition at the top, and then I'm showing you a difficult state transition at the bottom. And you can see the marked differences in the number of control signals needed. So for this easy state transition, notice that we're using the modular network again. And the first state is activation of the first 5 units and deactivation of the second. And what we want to do is to transition such that the first module becomes quiet and the second module becomes active. To allow this state transition to occur, all we need is a generic inhibitory control signal into the first module and a generic excitatory control signal injected into the second module. So this transition is easy because it only requires 2 generic control signals. The difficult transition I wanted to show you is in the bottom of the slide, where we have the same modular network as we had before, but now we have a different set of states on top of it. So here is this binary activation state: inactive, active, inactive, active, inactive, active. And what we want to do is anything that was off in the first state, we want to turn on in the second state. And anything that was on in the first state, we want to turn off. So we want to just switch every single binary variable here. To do that, we need an excitatory or inhibitory control signal injected into every single node in the network to allow that to happen and to minimize the flow of information from one unit to others. So this intuition, hopefully, that I've been building over the last few slides is one that we can crystallize and formalize in mathematics. And it's in the area of mathematics that's called network control theory. So network control theory is a subfield of engineering, systems engineering specifically, that allows you to study a network system and understand which control signals need to be injected into the system to allow a state transition to happen. And how can we quantify the ease and difficulty of that state transition? And how does it depend on the actual structure of the network topology in that particular system? So we've been working with Fabio Pasqualetti, who is a control theorist at UC Riverside, since about 2011, so about 10 years. Time flies. And with his collaboration, we've been thinking about how to take the beautiful mathematics in that community and bring it over into neuroscience. So the very first thing that we need to do in thinking about how to bring network control theory over into neuroscience to really quantify the ease or difficulty of state transitions is to write down a model for the way in which activity might flow through a nervous system. And I'm going to write down the very simplest model that you could imagine writing down here, but there are incrementally more sophisticated, more challenging, more complicated models that you could also write down, but I'm going to go for the simplest one for the moment. And so here it is, I write down X is the state of the brain at time t+1, and that is going to be equal to the pattern of connectivity between brain regions. So this A matrix is an adjacency matrix that indicates the strength of connections between all possible regions of the brain. And that typically in human neuroimaging is defined using what's called diffusion weighted MRI, or diffusion weighted magnetic resonance imaging, which is a type of imaging technique that allows us to extract which pieces of the brain are structurally connected by big bundles of neuronal axons. So that adjacency matrix is then multiplied by the state of the brain at time t. So far what this is indicating is that wherever the brain was at time t, that activity can flow along these structural connections to bring us to the pattern of activity at time t+1. So that's what we have so far. And this last term is the control input that's being injected into the system. So U sub kappa is the control input that's injected into kappa regions at time t. And then this B is a matrix, typically with ones along the diagonal, that indicate which regions have the control input being injected into them. So together, what this simple model does is it suggests that number one, activity can flow along structural connections in a neural system, and number two, there can be exogenous signals that could alter the flow of that information. So with this approach, we can now state how easy versus how difficult a state transition is. So conceptually, what we're going to do is to focus over here on the left-hand side of the slide, where X naught is a baseline state of the brain. Let's say it's activation of motor cortex. And then if no extra input was injected into the system, the brain might move along this gray trajectory over into this grayscale image called X sub capital T over on the left-hand side. But if control input is being injected into the system, then it might follow along this color trajectory from blue to purple toward a final target state, which is activation of visual cortex, for example. To quantify how that trajectory would happen, how much control input would need to be injected, what we do is that we build on that simple model of network dynamics I just mentioned, and then we define a cost function that penalizes the control energy required to allow the state transition to happen and the distance of the current state X from the target state. So let me just walk you through that really briefly. Here's the minimization, and we're minimizing two terms. This first term is the distance between the current state and the target state. So the current state is X, the target state is XT. And then the second term is the control energy being injected. Notice that there is a parameter rho out here in front of the energy term. That parameter can be used to determine the how important energy minimization is versus distance minimization in explaining the trajectory. So I want to sort of quickly motivate why we have these two terms in the minimization. So we want to minimize the energy being injected for a biological reason and for a clinical reason. So the biological reason is that there's a lot of evidence from the study of neural systems that there are constraints on the energy use and metabolism of the brain. And so that sort of constraint to minimize energy as much as possible suggests to us that we should be building models that have a penalty on energy use. The clinical reason is that when and if we are using this to try to understand exogenous stimulation of a brain in the context of, for example, quieting an epileptic seizure, we want to make sure that we use minimal stimulation so as not to heat the tissue. So there's both a clinical reason to minimize energy injection and a biological reason to minimize energy injection. Now I want to motivate the distance factor. So we want to minimize the distance of the current state from the target state because that allows for the trajectory to be relatively straight through the available state space, rather than allowing the brain or suggesting that the brain would move sort of haphazardly in a very high dimensional space before getting to where it needs to go. So again, that's kind of an efficiency assumption. I'm noticing that there are hands and questions in the chat. Should I go ahead and do that now or wait till the end?
Great. There is a question in the chat about examples of exogenous signals for control matrix.
So an example would be brain stimulation. We've used it previously in some published papers. In fact, I think I have it here. This Stiso et al. 2019 Cell Reports paper is where we use exogenous stimulation, grid stimulation, and there's an energy of that signal being injected into the brain.
Thank you. Bianca, do you want to turn on your microphone?
Yeah, I just wanted to ask, because in a way, distance is related to energy as well. Right? So a longer distance will require more energy than a shorter distance.
That's a really good question. So there can be multiple kinds of energy, I think is what you're pointing to. There could be multiple different kinds of energy. One could be the energy of the injected signal, and another one could be energy expended in walking, taking a longer time perhaps to reach the end point, or walking through a more difficult part of the state space.
More in terms of collinearity of the two variables, because they're correlated, right?
It depends a lot on the nature of the energy landscape. So which depends on the architecture of the network. So in general, they shouldn't, they don't, you can come up with cases in which they would not be correlated.
Okay, thank you.
Mm-hmm. Great question. So here, what I wanted to briefly mention is questions that we can then ask and answer with this conceptual and theoretical approach. So first, we can ask which brain states require least energy to go towards, or we can ask how much energy does it take to activate different cognitive circuits, like the reward circuit or motivation circuit. We can also ask, are the brain networks of some species easier to control than others or require less energy to drive to different brain states? And the answer is yes. If you're curious about cross-species comparisons, that result is in the Kim et al. Paper from 2018. Fourth, we can ask, could individual energetics be used to help personalize brain stimulation? We could ask, how do these energetics explain individual differences in brain-computer interface learning? So where some people learn more easily because the energy required for those state transitions is lower given their network architecture. And last, we can ask, how do we use these methods to ask questions about your own data? So we have a methodological paper that we recently published to kind of walk through how this all happens. But across these different types of questions, what I really wanted to emphasize is that each state change has an associated energy cost. And that associated energy cost is in part determined by the network architecture in neural systems. So that architecture constrains which of these transitions are easy versus which transitions are hard, requiring a large change in energy. The quick question in the chat is, could some of these concepts be applied to gene networks? The state would be the set of expressed genes. This is probably done in the literature, just a thought. Thank you for that question. I do think that there is one paper I saw very, very recently that started to use some of these ideas in gene networks. It hasn't been very common yet, though, so I think there's a lot more to do. And there's another question about how is distance in space calculated exactly. It's just defined as an L2 norm, so it's a very simple measurement of distance. So now I wanted to move into the second section of the talk to use the theory to probe normative development. And to motivate this section, I wanted to ask, what sorts of transitions are important for a brain? And you can probably all sit there for, even a second or less and think, imagine all the different state transitions that my brain needs to be able to do to allow me to engage in my world. It's going to be many, 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 right? So that motivated us to think, well, maybe we shouldn't necessarily focus on a single state transition right now. Maybe we should begin by thinking about whole sets of state transitions that may be important for a neural system. And so we went back to the engineering literature, and in that literature, there are two kinds of state transitions that people often talk about. One kind is where you move to lots of the easily reachable states on the energy landscape. And then a second kind is when you move preferentially to a really distant state on the energy landscape. So one is pushing you into a really distant state, and the other one is pushing you around what's accessible. So these two kinds of state transitions, I think, are very relevant for thinking about how the brain works, and I'll show you kind of why in the next slide. But I just want to mention that there is a way of calculating the ability for a region to enact one of these state transitions, given that model of dynamics that I showed you earlier. And given the time, I think I'm not going to walk through these equations right now, but I'm happy to discuss them more in the question and answer period. What I do want to do, though, is to show you that those two kinds of state transitions can be enacted well by different regions of the brain. So at the top here, what I'm showing you is a human brain, the sort of surface of a human brain, the lateral surface on the left-hand side here, and the medial surface, so in the center on the right-hand side. And then I've color-coded regions that are in what's called the default mode or resting state of the system. Those are in dark blue. And then in bright green, we have regions in the frontal parietal cortex. These are regions that are implicated in executive functions, which recall is what we're really trying to understand. So what we find is that regions in these frontal parietal areas that are known to be engaged when we have to enact executive functions, these tend to have high modal controllability, so MC, and relatively low average controllability, or AC. So what does this mean? What this means is that these regions that are implicated by a broad literature in transient cognitive control processes also are structurally predisposed based on how these regions are connected to one another. They're structurally predisposed to be able to push the brain into difficult to reach states far away on the energy landscape. And so we think that this is really interesting because it suggests that there's a structural explanation for why these regions become activated when you need to move between different tasks or engage in executive functions. And then on the right-hand side, what I'm showing you is the regions that are in the default mode system. And these tend to have high values of average controllability and low values of modal controllability. What does that mean? That means that these regions that are active when humans are just resting inside of a scanner, they are the ones that show they have a structural predisposition to be able to push the brain into most of the accessible states on the energy landscape. And I think that is really interesting because it suggests that the brain is located in some optimal place where those regions that are active at rest are the ones that have a lot of power to push the brain to where it's going to mostly need to go to throughout the day. So that then that observation motivated a follow-up question, which is, do these two kinds of control strategies change as children develop from childhood through to young adulthood? And why would we ask that question? Well, it's in fact over this very long developmental period that executive functions really come online, and it's estimated that executive functions don't really hit adult levels until about the age of 25. So this suggests that if the network control statistics are explanatory of executive functions, then they should also be changing over the same developmental period. So to address this question, we worked with Ted Satterthwaite, Raquel and Ribbon-Gur, and many others in the Brain Behavior Laboratory here at the University of Pennsylvania in studying what's known as the Philadelphia Neurodevelopmental Cohort. So this is a group of youth between the ages of 8 years and 22 years. All of them underwent an extensive cognitive battery, as well as a large imaging protocol. So every data point here is a different youth inside of that cohort that had the diffusion imaging data that I mentioned was important for estimating the structural connections between brain regions. So every data point here is a person. I want you to notice two quick things. One is that along the y-axis here is the modal controllability of that subject, and along the x-axis is the average controllability of that subject or participant. And you can see that across people, there's a positive correlation between the average controllability and the modal controllability. So what does that suggest? That suggests that brains that have high levels of one also have high levels of the other. So at least over the developmental time period, it does not appear that these strategies are competing or at odds with one another. It actually seems that they are complementary. I'll also want you to notice that the brain data up here has significantly higher values of controllability than two different common null models used in network science, those that preserve node strength and those that preserve node degree. So, but the key fact that you're probably wanting to know is, well, where are the 8-year-olds and where are the 22-year-olds? And what we find, this is the exact same data, just replotted so that the age of the person is indicated by the color of the data point. So here what you can see is that the brighter yellow colors, the older individuals, tend to have high levels of modal controllability and high levels of average controllability, whereas the younger kids, closer to 8 years of age, have lower values of both. So this indicates that these network controllability statistics increase throughout development over the same period in which executive function is coming online and reaching near adult levels. I'm going to finish this slide and then I'm going to go to the chat. So following up on this observation, we thought, well, this is at least some preliminary evidence to suggest that there might be a relationship between network controllability and executive function. There's this nice mapping between the modal control statistic and the regions that we know are activated for executive functions. And there's also a nice change in the controllability statistics over the same developmental period in which executive functions are coming online and reaching adult levels. But then we wanted to dig a little bit further, and Eli Kornblath in the lab published another study, which is complementary, where he showed that individual differences in network controllability are correlated with measures of behavioral impulsivity. So this behavioral, our ability to control our own behavior was correlated with the network controllability of the brain. So again, building this link between the network control profiles and the actual behaviors implicated in executive functions. So together, these results suggest that our theory is a useful marker of cognitive control, or how the brain enacts control to change network function. It looks like actually those are just about the recording. So I'm going to continue then on to just this very last section of the talk, where I wanted to use the theory to probe executive function and dysfunction, specifically in the context of schizophrenia. So to motivate this last section, I wanted to ask, how do we study not every state transition that the brain might need to enact, but one particular transition that may be critical for executive function? And that transition that we think is critical for executive function is activating areas in the frontal and parietal cortices of the human brain that are implicated in executive tasks. So here's an example of exactly what that state transition is. So here we have a baseline of the brain. And what we want to do is to model what energy needs to be injected when and where in order to drive the brain to activation of this frontal parietal circuitry that supports executive function. That energy is going to be different for every person because every person has a different pattern of network connectivity in the brain. And so what we're going to do is to estimate that energy for 946 youth, again, between the ages of 8 and 22 years. Here is the first result that we were really excited to see. And this is the control energy. So how much energy is needed to activate that frontal parietal circuitry as a function of age. So we hypothesized that control energies should decrease with age to allow people to activate this circuitry more and more easily, and that ease of activation would support the use of executive function in older youth. So we did indeed see this negative relationship that we hypothesized. So it suggests that the older individuals require less energy to activate that circuitry than the young kids who require more energy to activate that circuitry. So this evidence certainly supports, but doesn't prove yet, that this structural network inside of the brain develops in a way to ease the transition to executive function, thus facilitating the frequent use of this cognitive process in older youth. But another key question is, well, is this just a developmental effect, or is it really specifically related to executive function? Right? Is this sort of a byproduct, side effect of development, or is it key to explaining executive function specifically? To address that question, we turn to the behavioral data. So we had everyone inside of this cohort engage in what's called the Penn computerized neurocognitive battery. So it's a large set of tests that the participants would engage in that would probe their executive function, their motor skills, their social cognition, their complex reasoning, and many other domains. We applied a simple factor analysis to that data to extract one factor that really all of the tasks for executive function load onto well. And so that's what this executive performance measure is along the x-axis. And so we're plotting that after against the control energy after regressing out the effects of age. Now, notice again that you see a negative relationship here. What that means is that the people who have high executive performance are those that require less energy to activate the frontal parietal system, whereas those that have worse executive performance are those that require more energy to activate the executive system. So this relationship is again in line with our hypothesis that the energy required to activate the system is behaviorally relevant. As a sort of side note, we also found that this area in what's called the bilateral middle cingulate mediates the relationship between age and executive function. But the final bit of the story that I wanted to tell you is related to an acknowledgement. And the acknowledgement that I wanted to offer is that I've spent the last 40 minutes telling you that state transitions matter. But it's not just state transitions that matter. It is also the ability to persist in one state for a certain period of time. For example, to hold attention in a particular state or to maintain, commitment to this particular task. So holding a state is also really important for executive functions. If we were only able to transition and couldn't ever hold, we would be behaviorally compromised. So if we think about holding a state from an energy perspective, it's important to note that persisting in a particular state will also require energy if that state is not a minimum on the accessible landscape. So to understand kind of the energy of transitioning versus the energy of persisting in a state, Urs Braun was a visiting postdoc in the lab at the time and now has his own lab at the Central Institute of Mental Health in Mannheim, Germany. And he focused on a common task that is used to study executive function. It's called a working memory task, and it has two sections. One section is one that just demands attention, and that's called the zero back state. That's just where you respond according to what is currently on the screen. It just demands that you pay attention. It also has a second part of the task that's called the two-back working memory task, and that's where you have to remember the stimulus that happened 2 moments ago. So this task combines an attention state, really focusing on the now, with a memory state, thinking back to what happened 2 moments ago. And we know from behavioral studies of this task that the precise control of both state transitions between the zero back and two back and persistence in each of these task conditions is required for optimal performance. So Urs first estimated the control energy that was required to move from the simpler part, the zero back, up to the two back, which is more cognitively demanding, and also how much energy is required to move back down to the zero back. And what he found is that the transition up to the two back requires more energy than the transition back down to the zero back. So what this suggests is that our measure of control energy is tracking with the cognitive demand of the task. So that's really, really nice. It means that we have a measurement that is potentially a proxy for cognitive demand. But he also found another corollary to that fact, which is that the amount of energy needed to hold the simpler task is less than the amount of energy required to hold the two back, the more difficult task. And to show you that, I want you to notice that this y-axis here is called stability. That's just the inverse of the energy required to hold a state or to persist in it. And you can see that the stability of the zero back is significantly higher than the stability of the two back. Interestingly, you'll notice every data point here is a person, and you can really see a great spread of stability measures across individuals, suggesting, marked individual variability. We wanted to probe that individual variability a little bit more to understand if it was behaviorally relevant. And what we found is that greater stability in the two back state, so the people who could hold the two back without requiring much energy, also showed higher task accuracy. So they performed better on the task. So again, making this connection between the energetic requirements of the task and the behavior. All right, so this is the very last data slide that I wanted to show you, and then I'm going to broaden back out and ask for questions. So in the final analysis, Urs wanted to ask whether the control energy that we were observing in this task could be explained more deeply by underlying neurotransmitter behavior. So he particularly was focused on dopaminergic signaling because there's a beautiful two-state theory of dopamine function, which suggests that D1 and D2 receptors change the energy landscape on which the brain is moving in a particular way. So to probe the relevance of dopaminergic signaling for this control energy that we are studying, he used a randomized placebo-controlled double-blind pharma fMRI study where participants were either given amisulpride, which is a selective D2 antagonist, or a placebo. And what he found is that the control energy increased for both transitions. So from the less demanding zero back to the more demanding two back, and vice versa, if you're on the amisulpride versus if you're on the placebo. So that, and you can see that here. So placebo is in the dark blue, and then the amisulpride, the selective D2 antagonist, is in the lighter blue. So what that suggests is that yes, when you manipulate the dopaminergic signaling, you find a change in the control energy required for these state transitions. And to provide even more confirmatory evidence for the role of dopaminergic signaling, Urs also studied a patient population which is known for marked dopaminergic abnormalities, but also deficits in executive function and alterations in structural connectivity. And that's a population of people with schizophrenia. And so here on the right-hand side, you see patients with schizophrenia in dark blue, and then healthy controls in the lighter blue. And what we found is a greater control energy is required for the patients with schizophrenia to reach the cognitively more demanding two-back state than for the healthy individuals. So healthy individuals require less energy to get there. Patients with schizophrenia require more energy to get there. So this is again underscoring the potential relevance for dopaminergic signaling in our findings. So broadening back out, what we did over this sort of midday period was, number one, develop a theory for state transitions and their control. Number two, to use that theory to probe normative development. And then number three, to use a theory to probe executive function and dysfunction, specifically in the context of schizophrenia. But the take-home message, even if all of the data, doesn't stick inside of your brain, the take-home message is that the network architecture of neural systems constrains which transitions are easy, requiring a small change in energy, and which transitions are hard, requiring a large change in energy, with relevance, as we've seen, for development, which we touched on in both sections 2 and 3, but also for disease and drug, which we mentioned in section 3. So with that, I'd like to acknowledge obviously that the funding that has supported the work and the people who were involved. I tried to highlight them as I went along, but I'd love to stop here and see if there are any further questions. Thanks so much for listening.
Thank you so much for a really exciting presentation. We have several questions in the chat and a few hands that I can see. Maybe we could start with quadri-aid. What would be the effect of aging or neurodegenerative diseases on controllability?
Yeah, that's a really good question. So we have not studied older age at this point, and we also have not studied changes in degenerative diseases. There is a really nice study from Michael Breakspear's group showing changes in controllability in bipolar disorder. And then there's another study by Boris Bernhardt and others on showing changes in controllability in epilepsy, because that is a neurological condition. So those are some that are studies that are relevant, but I think that there's much more to do.
Puya, do you want to turn your microphone on?
Yeah, sure. Yeah, you hear me?
Yes.
Hi.
Yeah, hi. Thanks for the talk. It was a great talk. A lot of stuff to process. My main question was about I think it was the research that you presented, one before the last, that you had this diagram of different stimulation at different points, and you were talking about activating the frontoparietal network. My question was mostly conceptually. I was asking, like, what does it mean, or what do you think it means to activate that network? Does it mean that you going to force your subjects through stimulation to take hold of their thoughts, or like, what does it even mean? Yeah.
Oh yeah, that's a really good question. In that particular case, what we're thinking about is what energy is required by the biological system to move to a different state. So it would be an intrinsic thing. We naturally activate frontal parietal cortex when we need to engage in task switching or decision making or a working memory task. So the question is, how much energy do we need to expend when we do that? You might ask then, what is the relationship, like what kind of energy is being used? This is a common question that people ask. And we have a paper that we just submitted, and it should be on bioRxiv hopefully today or maybe tomorrow, depending on the checks that they always do, but where we connect the control energy estimates that we have from this theory to and estimates of metabolism available from FDG PET. And so I'm really excited about that because it provides some more biophysically, more connections to the biophysics of like what energy really means. Because in the theory itself, the time is in arbitrary units, energy is in arbitrary units, right? It's a very abstract sort of phenomenological model. And so connecting it to the biology as available from FDG PET, I think is exciting.
Thank you. Can I maybe just go to Yasser now, and then we'll go back afterwards to the chat.
Yeah, thank you, Dani. Something that is considered in the control theory field is that the validity of the signals that are identified to control the system depends on the underlying model, on how accurate is the model to represent the dynamical system that we are modeling. You have used it here, like in many different, trying to reply many different questions, the most simple linear model. And then my question is, how well do you think that this model will reproduce neuronal activity in a complex system during some time? And in addition, is some of the conclusions or findings that you have been observing could change when a more complex and realistic model will be applied?
Yeah, those are really great questions. Thanks for asking. So the simple linear model that I showed here is certainly a very gross approximation of the neural dynamics that we know exist in nervous system. So we know that there are lots of nonlinearities and lots of complexity in the dynamics. So the linear model should be thought of as an approximation around an operating point of the system. And the question of sort of what the operating point is, what the time scale is over which this would be useful, what the spatial, even the cognitive space over which it would be useful, those are all questions that I think require further study. One point I do want to make is we've moved more recently to a time varying model instead of where the adjacency matrix itself is time varying. So this moves beyond, it adds a nonlinearity in the connectivity. And so we just have a paper that was published in the Proceedings of the National Academy of Sciences. I feel like it was a couple months ago, so relatively recently, where we study seizure dynamics and we extract patterns of effective connectivity in different parts of the seizure and then ask how the control properties of the system change in different parts of the seizure. So the idea being that structural connectivity is a fine, generic guess at which connections are relevant, but for any specific dynamical context, you might want a very precise pattern of connectivity that was used then. And so that's what we've been using more recently. And I'm really excited about taking that, which we applied in a clinical context. I would like to use that more in a cognitive context as well, because as we move through different cognitive tasks, we may be using a very different pattern of connectivity, effective connectivity between brain regions that would alter the control predictions.
Alan.
Dani, wonderful talk. I would like your perspectives on what is the optimal scale at which to consider these questions, taking into account the spatial resolution of the different imaging modalities that are involved. Should we be dividing the brain up into, 100 regions, 1,000 regions, or 1,000,000 regions? And how stable are the modeling results as you vary across that scale?
Yeah, that's a really good question. In most of the studies, we do try to apply multiple parcellations that are across different scales from, let's say, 100 regions to 500 regions, just to show that there's consistency across those parcellations, or consistency in our results across those parcellations. But in some sense, I feel like that's a practical answer. It's not necessarily a very helpful first principles answer. So I think to me, at least the way I read the field right now, is that the jury is out about whether there is a right scale at which to be coarse graining the brain, or whether we need to be understanding more about how the dynamics or how the structures change over multiple scales. So our questions are multi-scale to begin with. I think that there's more to do. I personally have become more and more interested in studying some of these factors at the voxel scale, just because we actually have that data available, and we're typically averaging over all of that information. Maybe we're losing a lot that could be useful for understanding neural function. So we've been doing a little bit more at the voxel level now, but yeah, I think you'd probably get a different answer from every neuroimager, right? So that suggests to me that there isn't necessarily a right scale. I think that there's a right scale for the question, but there are so many questions that it's important to maintain diversity in our approaches. It's my take.
Thank you. I think we have time for a few more. Dominique Boutet, do you want to unmute your microphone?
Yeah, I can ask the question verbally. I was wondering how we could relate synaptic plasticity to those networks, because those networks, I would assume they're not just entirely structural, because the strength of the connections is dynamic and can be changed with time. So I was wondering how we could relate that to, improving task performance, in the sense that if we improve the connections, we can increase or decrease the energy cost of a state transition, which would make a task easier to perform or a state easier or harder to maintain, and how that could relate to the idea of memory.
Yeah, that's a really, really interesting question. So we haven't thought about it necessarily in the context of memory exactly, but we have thought about it in the context of habit formation. So I have which is connected. So I have a collaboration with Anne Graybill at MIT and Teresa Desrochers at Brown University, and they study macaque behavior and neurophysiology, and have really well-developed programs in understanding learning and habit formation in macaque monkeys. So there we did study the formation of habitual behaviors and demonstrated that the energy required becomes less for those behaviors, for those state transitions, supporting those behaviors becomes less and less with time. So it suggests an energetic explanation for habit formation. Now this isn't exactly memory, but I think it's connected conceptually in the sense that what we may get by task performance is the capacity to reach states with less energy. And that may be supported by something like synaptic plasticity. So I love your question. I think there's a lot more to do here.
Paul Cizek, there's a bit of a conversation going on between Paul and a couple of other people, but Paul, do you want to start off?
Sure. Hi. Well, no, my question was really about kind of related to Alan Evans's question about the scales, that it seems that there's an implicit assumption here that the state of the brain is well captured by the state of activations in some kind of regional space or even voxel space. And that therefore implies a kind of assumption that the important states are not related to the pattern of all the neural activities within all those voxels. And is that an assumption you're making?
That's a really good question. I'm really interested in this, at least in this talk, in understanding the pattern of large-scale activation that we know accompanies executive task performance in humans in an MRI machine. So my question is really to understand that pattern of activity, how it differs across people, how energetically expensive it is. So in the context of that question, I care a lot about this large-scale activation. However, there are other studies that we've done, but also other studies from other labs that have used the same network control approaches to understand cellular level activity. And I think, I think that both there are important and interesting questions at both scales, and I'm not trying to suggest that one is more important than the other.
Are there examples where the same question has been asked at both scales, and are there answers congruent?
That's a good question. The study at the cellular scale that I know most well is from Barabazi's group, and it's evaluating motor control in C. elegans. And I haven't seen that specifically done in humans. That would be an interesting comparison, though.
Thank you.
Maybe we'll take one more question if somebody has one. I think I missed one in the chat from Nilufar. Nilufar, are you still here?
Yeah, I'm here. Just my question was that how do you compensate the effect of noise in estimation of the next state of the model? Because yeah, it's not just an input that you receive from external, it's other effects. That was my question, and how to estimate it, how to compensate it.
Yeah, that's a really good question. At the moment, the current model that I showed you at least does not have noise included, but it's something that you can add either in simulation or to the actual formulation of the model. But at the moment, that's not something that we explicitly estimate. So I think that would be a neat way to move in the future. I think if we're talking about noise, we should also talk about uncertainty. And there is very clear uncertainty in the estimates of structural connectivity that's acquired from the diffusion imaging signal. So I also think that's another place where we could add more rigor, is to have those estimates of connection strengths have actual, distributions and levels of certainty in them too. So, yeah.
Thank you.
Mm-hmm.
So I think we're past the hour. I would certainly like to thank you, Dr. Bassett, for a really accessible talk, appealing to all of the people who came and joined us today. Very thought-provoking, links to all kinds of other domains, stimulating really exciting questions from the group, and and certainly making me think about interesting ideas for questions to follow up. So thank you so much.
Thank you so much for having me.
Presentation. I'd like to make a quick note to the quantitative life sciences students. Just stay online at this link for some time with Dr. Bassett. Starting basically now. We can give Dr. Bassett a few minutes break, but otherwise we'll just stay here on this link. And thank you all for attending today. Would you like a really short break, Dr. Bassett, before we get going with the students, or are you okay?
I'm good. I'm good to go.
Okay, wonderful. Alan, I know you have a different link established for later on.
Yeah.
Okay.
I'll talk to you at 2 o'clock, Dani.
Great. So you have some students, as you can see, in the seminar room, a few here, and others that are online with you. And Bianca, I know, had some questions. And anybody else? Christian also? Take it away. Do you guys want to introduce yourselves? Turn your cameras on.
Celia, should we start with the room, with those in front of us in the room?
Go for it.
If we could just kind of position ourselves towards them and say hi. So Dylan, you want to start?
Yeah, sure. Name's Dylan, first year QLS student. We'll be working in neuroimaging, but also trying to bridge in some genetics. So